Thank you, and thank the organizers for the kind invitation and opportunity to share our recent results in a proof of concept run of ONT long read sequencing technology in the clinical setting. Rady Children's Institute for Genomic Medicine was founded in 2016, and at that time was only due to a very generous, huge philanthropic donation from the Rady Foundation, because there are multiple obstacles for the really sick kids to get a molecular diagnosis, ranging from the physical availability of the labs and the insurance coverage. The Rady Foundation covered for all of that. We are embedded in the Rady Children's Hospital, which is a large regional hospital serving 3 counties in Southern California, the only level 4 neonatal intensive care unit in these 3 counties, and provides healthcare to a lot of kids' visits per year. Most of our patients are under 3 months of age. The mission of our institute was stated as ending diagnostic odyssey for the critically ill kids and newborns. And we established a clinical lab and clinically validated, CAP-clear certified. We expose a clinical test of rapid whole genome sequencing, and we have been running it for 7 years in increasing volumes, reaching 15,000 sampled cases by now. And that is where we drew the foundation for trying some next steps. Only 35% of our kids get a diagnosis, and that's not because we're particularly bad at it. It's comparable to the other centers' stats, such as Mayo, Baylor, and so on. So it's depressing to think that 2/3 of our kids walk away with continuing their diagnostic odyssey, even though we know that, strongly believe that they have genetic etiology for their condition. So the collective we want to do better than that. And we see a huge promise in the long-read sequencing technologies such as ONT. Long reads are good for a lot of things, but we will focus on methylation only. In consultation with our attending physicians, we chose some representative diverse cases, and it was 11 cases with known molecular findings, including some with known methylation dysregulation in them, and 42 previously undiagnosed, one set of healthy parents to form an embedded one complete trio. So at the risk of telling you what methylation is for the 5th time in the span of 2 days, I will be very fast. The methylation of the 5th position of cytosine is the most common modification of mammalian DNA, occurs in cytosines followed by guanines. The CpG dinucleotides cluster in specific regions of two varieties, one in the very repetitive regions, and they are fully methylated there and are of no interest. And the second one is in the CpG islands that collocate with precise transcription start sites of genes, and this is where they're mostly constitutively unmethylated, and that's where they control if the gene would be silenced. We have 2 chromosomes, one from mom and one from dad, and almost all of them, the CpG islands for them are unmethylated, and the expression is unrestricted, with the important exceptions on chromosome X. Females have 2, so they randomly silence one of them, and this calico cat has random patches of skin and coat, because the coat color is encoded by gene O, stands for orange, and it's indeed located on chromosome X. Her male kittens might have full orange coat or fully non-orange coat, but not calico. And so outside of chromosome X, some of the genes have very specific pattern of origin, cell-specific methylation that starts at the beginning of the new generation cycle in sperm and eggs. No matter where the chromosomes came from, they will be methylated in the pattern specific to this cell. Some genes are silenced in the egg, some genes are silenced in the sperm. This leads to a sudden expression of previously silent variants. Imagine a family where grandfather is, and grandfather and father and his daughter are all totally healthy, but unknowns to them, they're all carriers of paternally imprinted, therefore silenced, pathogenic variant, and that only becomes evident by the kids of that woman. Uniparental disomy is another effect that leads to unusual non-Mendelian effects in expression of genes, and that happens when both chromosomes, due to meiotic error, are inherited from the same parent, and that's all. And that's some of the cases that we want to be able to automatically catch with minimum time expense, because our interpretation team has to process dozens and hundreds of cases per day. They cannot look at all of them. This is how the methylation looks in the familiar IGV browser. We have 2 genes here, and we have 2 samples here. The bottom one could be replaced by any other of the 53 samples. We see that the gene on the left is universally unmethylated in its CpG island, and it will be unrestrictedly expressed. Not so for the gene in the center. The bottom quarter, the haplotype 2, and we know from additional data that this is the paternal haplotype, paternal parent of origin haplotype, is unmethylated. This allele will be expressed. The other one will be silenced. And indeed, this gene is aptly called PEG1, which means paternally expressed gene 1. Not so in the sample above. This is, you can see 2 features. All of the reads are methylated. None of the expression from this gene will show up. And why is that? This is because this is maternally ISA deployed genome. Both of the chromosomes, and they're identical, were inherited from mom. No expression for this gene, and some genes to follow on chromosome 7, and that leads to the phenotype of Russell-Silver syndrome. We aim to be able to, register these deregulation and methylation automatically. We used to have many, moving parts in this pipeline, which is now greatly simplified by latest releases of epitome human variation workflow, that haplotypes with WhatsApp, that quantizes the methylation probabilities with modkit, and then we further summarize them, as I will show in the next slides. In half of our slides, we happen to have supporting documentation, data from prior retrospective data for the parents, and from them we only need SNPs. With SNPs, we can de-identify haplotypes 1 and 2 to their correct names, maternal and paternal parent of origin, and then we follow up with the same system. To quickly find outliers, we construct a background of normal methylation patterns, either in 1000 base windows or in predefined CpG islands or other user-defined regions. And we can appreciate that here, only 12 samples are shown. In every other line, we can see high methylation or low methylation. These are pairs of haplotypes from representative samples. We statistically summarize them into averages and standard deviations, and we fill up an object of a panel of normal methylation based on a certain training material. Then if in some future clinical lab, we could use it for a while, just like people do that with predicted structural variants from exomes, which is hard if you don't know the panel of normals. In our case, of course, we bootstrap, we go back. In the past, well, I did mention that the top sample was Windsorized. Windsorization is where you throw away the outliers on both sides, which is commonly known to people who watch figure skating. The outlying scores are discarded. The top sample will be highly rated in this particular location because it has significantly hypermethylated the allele that was expected to be low methylated. If we arrange all of the scores in a volcano plot, we can appreciate that it is well behaved. If we tally up all of the outliers and group them by samples, we recover the Russell-Silver syndrome case that I previously described, and it is topped only by Down syndrome, which is when it was included not for recovering Down syndrome, which is embarrassingly easy from any molecular diagnostic labs, but for coinciding other variant. But we recovered the well-known published effect that in Down syndrome, there is a significant methylation deregulation, and not on chromosome 21, but on other chromosomes, 5, 10, 12, and so on. So in conclusion, I would like to say that methylation, we expect it to be very important for a boost in diagnostic rate in pediatric genetics. And conveniently, ONT provides a way to find genetic variation and methylation in one go. And we hope that the software will be made public soon. We're preparing it, just clearing it for legal reasons. And I would like to acknowledge the rest of our lab, the esteemed collaborators at Oxford Nanopore, and our wet lab, who worked hard to make long read libraries. And I thank you for your attention.